Okay, in this video we're going to talk about the disease known as sickle cell anemia. And sickle cell anemia is caused by a change in the shape of your red blood cells. So red blood cells, as we can see here, these are red blood cells. So here is a normal red blood cell, and here is one that is abnormally shaped. Now you can see in a regular red blood cell there is a little dimple here, and that's because this red blood cell has lost its nucleus. And you're going to say, wow, how can you lose your nucleus? And why would you lose your nucleus? Well, red blood cells are designed to do one thing, and that's to carry oxygen through your body. And essentially what they want to do is there's a chemical called hemoglobin that carries that oxygen around to all your cells. It also picks up CO2, which is a waste product from your cells, and carries that out of your body. It carries both of these, it carries them to and from the lungs. And the idea is it's a little package of hemoglobin. And the cells, as they mature, they make as much hemoglobin as they possibly can. They pretty much turn off all other genes, and they just leave the genes on that make hemoglobin. And with that, then they want to package them and pack as much in there as they can, so they actually more or less die, and by destroying their own nucleus to pack more hemoglobin in there. And then they're released from wherever they're made. They can be made in a couple of different places, in the bone marrow primarily, but there are other places they can be made. And where they're released into your body, and that's all they do. They last several weeks, and they just transfer oxygen and CO2 in and out of your body. And this is the normal shape. It has a little dimple, and that dimple is formed when the nucleus actually is taken out, or is dissolved, I guess is the way you might say it. But it essentially, that nucleus made as much hemoglobin as it possibly could before it was destroyed. Now in the disease over here, there is something occurs and causes the shape to change. This shape is more or less rounded, and because of that rounded kind of shape here, they flow through your blood vessels pretty well. They bounce off of each other more or less. But with this shape, they don't bounce off each other, and they seem to form, they get stuck in the where the arteries and veins branch. They kind of get stuck like grains of sand in a small tube, and they clump. And that clumping, of course, blocks the flow of blood, and therefore people can die. They get blood clots from these just kind of not being able to pass through the smaller blood vessels. So it's a very bad disease if you get it. And the reason that these form these shapes is because when you have a lack of oxygen, the hemoglobin changes shape. So the hemoglobin is a protein, all right, and that's what they mainly carry. And when it runs out of oxygen, so you've been exercising a lot, and there's not a lot of oxygen to put into these cells, the proteins change shape. And when the proteins change shape, they stick together, and the proteins become sticky to one another, and they form long rods. And when they form these long sticky rods, the shape change of the whole cell changes. And so this is a very bad disease. It does have some advantages, though. It gives you a kind of semi-immunity to oh, I've forgotten the name of the disease. You get bit by malaria. So malaria, if you live in an area where there are mosquitoes, you can get malaria, and this gives you a resistance to malaria. But it also means you can't exercise much because you'll create blood clots in your body. So how does this work? Let's blow this one up a little. This one doesn't have very good graphics anyhow. Let's scroll back here. Here we go. Oops. The idea here is, I'm just demonstrating here with this really bad picture, that here's how they normally flow through the blood vessels when you get to a branch. But when they're in the sickle form, they will form this little clot, and you can see that even the good ones that are left can't get through. There's nothing. These little clots here. And of course, if you can't get blood through your body, it's going to kill you. Now here is the way it looks in a normal cell, where all of the little hemoglobin molecules are separate. It's hard to see here, but you can see that the little dots are separate. But here, the dots are not separate. They form long chains. They stick to one another, and this causes this sickle shape of the cell. All right, now what causes it? It's a very simple mutation. If you've done the lab 0, you will understand what's going on here. And all that's going on is that there was a mutation in one of these codons. Now, if you don't remember what a codon is, go back and rewatch the labs. And here you have a T, which codes as an A. Here you end up with glutamic acid. All right, so a T in the DNA, and that would be an A in the RNA. They didn't change it to U's, but that's okay. And you end up with something called glutamic acid. It's one of the amino acids. But with the mutation, you have a mutation from a T up here to an A here, which reverses it. So in other words, it's kind of like you reverse these two, but wherever you have a T, you have an A on the other side. So if you change this side to A, this one's going to be a T, and actually it's a U if you look at the genetic code or whatever. And it ends up with valine instead of glutamic acid, and that single change causes the hemoglobin to be sticky, and therefore all the little molecules of hemoglobin inside the cell stick to one another, and you get this shape change. So normal cells, and here are the sickle cells, and they of course cause clots. So here is a little diagram. Let's see, can we blow this up just a little? No, let's leave it this size. So you have the gene. Here's the gene on the DNA, or this would be a gene here, and you have two bases, the A and the T here, and there is a mutation. Something has happened, and therefore the protein, it doesn't work right. The protein is shaped wrong, and it ends up being sticky to itself. All right, and if you inherit that trait from a parent, or you got the mutation yourself sometime in your lifetime, then you're going to produce this sickle-celled shaped red blood cell. And if you have those, then you get blood clots. Now, luckily, this is a mutation that occurs in many people. It doesn't kill them right away, because the only reason that it would become sickle-celled is when there's a lack of oxygen. So a lack of oxygen, like you're exercising really hard, you pulled all the oxygen out of your blood, and what that does, that's when they become sticky. They don't necessarily become sticky under normal circumstances, only when you really exercise hard. So these people, of course, are pretty much reduced to not exercising much, and they do not live long. They live into maybe their 40s at the most. I think these days there are drugs and more treatment to help them live a little longer, but traditionally they lived into their 30s, maybe their 40s, but not much longer than that. But you diagnose this today, you can live much longer. And if you only have one copy of the gene, remember you have two copies of each of your genes, and you get one from the mother and one from the father. We're going to learn about that in the second exam. And the idea here is, if you only get one copy, you're not too bad off. You may live a little longer, but if you get two copies, you're going to be sick pretty much your whole life, and you're not going to be able to exercise at all. So I just wanted you to understand how this one worked, kind of an idea of how genes relate to the proteins. You change the gene, you change the protein, and that protein can lead to a disease. And this is a very simple version of how a change in the shape of a protein that makes it sticky will allow a disease to occur. All right,